Salvinia species. Salvinia is a water fern. It's an aquatic pteridophyte that is much old. It is fossils of this Salvinia has been found in the rocks during the Cretaceous period as well as during the Tertiary period. So we see that this is an ancient aquatic pteridophyte, and in this part of the video I will be discussing about the reproduction and life cycle in Salvinia species. Hello friends, I am Ayantika and I have started doing videos on MSc and BSc syllabus. I have already made videos on algae, fungi, bryophytes, the lichens, cyanobacteria, and now I have started pteridophyte, of which I have made videos on the general introduction of pteridophytes and another water fern, that is Marsilia, and all the links I have put in the description box. The links of the pteridophyte videos, and you can go to my channel to watch more videos on other topics. So now let's start the video on reproduction and life cycle. So, reproduction in Salvinia is of two types, especially in species, for example, Salvinia auriculata, where the spores are not viable. So, the reproduction method by fragmentation, it is the only method by which Salvinia auriculata, which is sterile, it has got a sterile spore. So in that case only fragmentation is the only method of reproduction. In other species of Salvinia, this is a very quick method of regeneration, and apart from this fragmentation, it also generates spore and it reproduces. So fragmentation mainly, this is the very young rhizome, and this fragmentation of rhizome at any parts, say for example mostly the nodal region. So this fragmentation of the nodal region or fragmentation of a branch, it usually occurs in all the species, and each of this new fragment, it develops into a new plant. Apart from that, when it reproduces by spore, here in this picture we see that the spores, they are born in a row or in cluster. Here it is born in a row on the surface of the submerged aquatic leaf. We know that Salvinia has got two photosynthetic leaf on the surface that is floating, and one submerged leaf that is much filiform, and each of the segment contains this sporocarp. Each of this yellow color yellow or golden colored, this globular region, these are the sporocarp in Salvinia, and each of the sporocarp contains inside it many sporangia. So it is a heterospora species, which means that two type of spores will be produced, macro and microspores. So it has two type of sporangia, microsporangia and megasporangia. So the spores in Salvinia, it is born inside sporangia, which is in turn born inside the sporocarp. Now in the previous slide I was showing that this golden colored globular structure called sporocarp, they are arranged in a row on the submerged leaf. So each of this sporocarp, inside it contains either megasporangia or microsporangia. We know that Salvinia is heterosporous and two type of spores are present, microspore and the megaspore. Microspore is actually ultimately give rise to the male gamete, and the megaspore give rise to the female gamete. So this is the picture. In this picture we see that figure A, it contains the transverse section of a sporocarp with microsporangia. We see that this sporocarp has got a two or three layered outer wall which is protective in function, and the microsporangia are born inside the sporocarp on branched receptacle. We see that these are the branched receptacle which contains sporangia on its tip. And this branch receptacle, mainly they are provided with vascular bundle and they provide nutrient to the growing sporangia. Similar is the case for the megasporangia. The megasporangia are also, we see that the megasporangia also borne on branches, but in some species megasporangia, they are clustered together without any receptacle. Whatever be the megasporangiate or the microsporangiate, the sporocarp, the megasporangiate sporocarp is born earlier. They are formed earlier, and they are formed in much lesser number than the microsporangiate sporocarp. The sporocarps that contain microsporangium, they develop later, and they are developed in much smaller size and much larger in number as compared to the megasporangiate sporocarp or female gamete producing sporocarp. So there are two type of sporocarp, megasporangiate and microsporangiate, and each of this sporangia, inside the sporocarp, the porangia, they produces the spores. So the megasporangiate sporangia, it produces, say for example 4 to 25 megas sporangia. The sporocarp contain 4 to 25 megasporangia depending upon the species. Of this 25 or 4 megasporangia, each of the sporangia, they develop into 8 megaspores. 8 megaspores. Ultimately, out of these 8 megaspores, only one survive, and rest of the megaspore, they just break down and add to the nutritive fluid, that is along with the tapetum. We know that the spores, it has got many layers of wall. The innermost layer is nutritive in function and is tapetum, and so the tapetum plus the degrading megaspore, all these two, they add to the nutrient content inside the spore, and this tapetum and the megaspore plasmodium that is formed, that is together, it is called perispore when it becomes much trabeculate. That is, it forms a vacuolated structure, that is called the perispore, and this entire structure is present inside the female, that is the megasporangia. So again, the megasporangia is composed of the 2 to 3 layered wall, and each of this sporangia I am talking about, here we see that each of this megasporangia, that is, we are talking about each of these megasporangia, this structure, it consists of 2 to 3 layered outer wall. The outermost layer, it is hairy, and the outer and the middle layer, it has got a vacuole in between them, and the innermost layer, it is tapental in origin. That is, after a certain stage, this inner layer, they disintegrate and add to the nutritive fluid, and inside this 3 layered structure are the spores, and 8 megaspores are formed, of which all but one survive, and rest of the disintegrating megaspore, they add to the nutrition of the single megaspore. And this megaspore, they ultimately, out of this 8, only one survive. So this single megaspore, it forms a single celled binucleated neck, a single celled venter, and a single celled large egg cell. Single cell large egg cell. So this is the structure of the archegonia, which contains the egg, and this is the ultimate structure that the single megaspore form. Now when we come to the microspore, so we see that the microsporangia, they have got numerous microspores. Generally, initially 16 microspore mother cell, they divide to form 64 microspore, and from this, from the microspore mother cell, 16 microspore mother cell, they form 64 microspores, of which they undergo meiosis to form 4 spermatocyte or the sperm. So this is how it occurs, and the the microspore also has many layered jacket inside which the spores are born. Now this entire the spore, from the spore form, the gametophyte generation starts. So once the gametophyte generation, that is the spores, the gametes, they are the gametophytic generation or the haploid generation. So the sporocarp, when it gets disintegrated from the leaves, the submerged leaves, when the sporocarp, they get separated from the leaf, the floating the submerged leaf, then the sporocarp sink to the bottom of the water. So once the entire sporocarp sinks to the bottom of the pool or water pool, so then the wall of the sporocarp, they gradually gets decomposed, and once the wall of the sporocarp is decomposed and the spores are liberated, the spores once liberated, they float to the surface of water again. So the before the spores are liberated from the sporangia, already once they are released, so this is how the spore looks, and this is the megaspore. Already they have undergone a developmental process. This is the microspore. That is this is the microsporangia. The spore has started to produce the spores. They are released and the to the top, and the so what happens at that time? They started to produce the megaspore. So once the megaspore already has formed this archegonia, and the megaspore, it floats on the surface of the water body in a horizontal position, this position. So this is the water surface, and megaspore are floating, that is the archegonium is floating. This is not the entire structure, this is the structure. So the archegonia is floating. This is the gametophyte, the female gametophyte. So this is the archegonia, and this one is the egg cell. So they float on the surface of water in a horizontal position, and the microspores also release, they float on the water surface, and the wall, the antheridial wall, it gets disrupted, and the sperms, they are liberated. Now once the sperm are liberated, they swim to the surface and reach the archegonia, the neck of the archegonia. The neck of the archegonia contained a single neck canal cell, a single ventral canal cell, all of which at maturity they got disintegrated into mucilage, and this mucilage in turn attracted the sperm to swim towards the egg. So the sperm reached the neck region, and then it fertilized. So after fertilization, what happens is that the zygote is formed. So zygote, though we know that the zygote lies inside the gametophyte, the archegonium. So in turn the zygote, they develop, they divide several times, the zygote, and the sporophytic generation. Zygote is the sporophytic generation, that is the diploid generation. Now so far I was talking this entire spore from the spore, that is from the spore structure, from the spore structure from the spore structure to the gamete, to the gamete region, that is the this is the female, this is the haploid generation. All this entire stage, that is from the spore to the gamete, through including the gametophyte, this is haploid in nature. But once the gamete, that is the sperm and the ova, they fuse, the sperm and the ova, they fuse to form the zygote. The zygote is the diploid phase. Zygote is the diploid, that is zygote is diploid, that is 2n. So the zygote further divide to form the entire sporophyte. So from the zygote to the entire sporophyte, to till
So,
the again the sporocarp,
this is
entire
about the
stage is
megaspore, how it is
sporophyte.
developed.
The we know that these are the tetrads of the microspore, the microspore tetrad. This is about the development of the microspore. We see that this is the megaspore, how it looks, and that this is the perispore which I was talking about. So the perispore, it consists of both the tapental as well as the disintegrated megaspore, and look how it is, a very highly vacuolated structure. And this structure, this entire structure in the apical region, this entire structure, this reminds us of the pollen collection region in the gymnosperm. So this is entirely formed of the perispore. So this microspore tetrad and this megaspore, this megaspore and the microspore, they are the sporophytic generation, and they ultimately give rise to the antheridia,
So,
archegonia,
the
and
zygote, which is the
ultimately
first
cell of
the
the sporophytic generation.
the
Zygote is a
gametes,
single
that is
cell,
the egg and the
sperm, diploid in nature, so undergoes division into apibasal and hypobasal cell. The upper cell, that is the apibasal cell, it again ultimately give rise to the leaf as well as the stem, that is the rhizome, and the apibasal the hypobasal cell, the lower cell, it give rise to fruit, as there is no root in the Salvinia species, so the hypobasal cell, they give rise to only fruit. So this fruit is a basically they have the function of the suspensor cells, that is they absorb the nutrient from the gametophyte for the growth of the sporophyte. And as soon as this cotyledonary leaf is formed, then the function of getting nutrition, it is formed by the leaves. So in this way, zygote develop into apibasal and hypobasal cell, and ultimately this type of structure is formed. here we see that this is how this entire developing embryo looks like. This is the final product, where we see that this is the leaf, this is the leaf, and this is a rudimentary root. As we know that there is no root in Salvinia, so this will soon be lost. And this is the stem, this is the stem region, the column. And this is not the part of the gametophyte, this is not the part of the sporophyte, but this is the gametophyte. We know that gametophyte, they had a this structure of the male gametophyte was like this, where the apical region, this apical region was it contained large number of cells, the apical region, where the archegonia was formed. So for example, these are the archegonia that is formed in the apical region, and this is the region which contain many free nuclei, and this is the basal region, and the apical region is cellular, and it contained the
life cycle in Salvinia.
archegonium. We know that Salvinia, along with other pteridophyte, they have got alternation of generation.
So
they have got a sporophyte that is sporophyte, and that is
this entire
this is the
entire
life cycle of a fern. Though we are dealing with only water fern,
So this
but
is how the young Marsilia
main
is
plant
growing,
body, that is
this
out
one.
from the gametophyte, this one here. So this is the plant body of Salvinia. So the plant body, this is the sporophytic plant body, and it bears spores. So this is the sporocarp, that is a leaf containing. But in Salvinia we have a sporocarp, that is a sac-like structure, and inside which the spores are born. As because it is heterosporous, that is two type of gametangia will be found, that is two type of gametangia will be formed. This is one, and another type of gametangia. Two type of gametangia will be formed, and two type of gametes will be formed. So as the gametes, they fertilize to form a zygote. So this zygote again, they give rise to the sporophytic plant body. This stage, that is the protonema stage, this is not found in Salvinia, but however the entire stage, that is the alternation of diploid and haploid generation, it is more or less same. So we see that in Salvinia there is a there is a sporophytic diploid plant body which give rise to the haploid spore, that is haploid sporophyte, and later the spore. The spore forms the gametes, and the haploid gametes, they fuse to form the diploid zygote, which divide and divide repeatedly to form the sporophytic plant body. So the plant body is sporophyte, and that is quite dominant, and the gametophyte, they are so small that only you can observe it with a hand lens. So this is the picture of the gametophytic sporangia. This is the sporangia which contains spores, which will ultimately form the gametophyte. So this was all about reproduction and life cycle in Salvinia. And in case you haven't watched about the anatomy and the external structure of Salvinia, or its general feature, its importance, and fossil of Salvinia, you can watch it in another video, the previous video, the part 1 of this video, and all the link of that video I have put in the description box. And I have made other videos on other pteridophyte also. All the links also I have put in the description box. And if you like this video, do hit the like button, do share it with your friends, and Also to subscribe, because your subscription, it encourages me to make more such videos. I am making videos on the BSc and MSc syllabus. So in any case you are interested, or you are a BSc MSc student or interested in these topics, do subscribe to my channel to get access to more such videos. Thanks again for watching my video.